Okay, welcome everyone to this one of the 4 parallel sessions for our afternoon Canadian Computational Science Spotlight. This is Arthur, who I've just brought up. So we're going to do things, we're going to be a bit more relaxed about this session than we have been so far. And specifically what I mean by that is we're not doing a kind of pre-presentation preparation. So all of the speakers, just so you know, I'm going to invite you up one by one, and I'm just going to ask you to start presenter view and give your talk, and then I'll tell you off if you're going over time, and then we'll move on to the next talk. Good?
Good.
So without further ado, I'm going to introduce Arthur. Actually, Arthur, I'm going to let you introduce yourself and take it away.
Okay, thank you, John. So I am Arthur Powanwe from the University of Ottawa in the London Lab. So I will present my work, Dynamic and Statistic of Gamma bursts. Let me start. Do you see my presentation?
So you just need to choose share screen. If you hover over your part of the screen, you'll see an option at the top that's share screen.
Okay. Okay, share. Oh. Well, let me. I don't see that option here. Yeah, I don't see the option share screen. I see share, but I don't know where is share screen. They share on Twitter, share Facebook, something like that. But
so when you hover over,
there's no share screen.
And there's toggle video, toggle mic, and there should be like a little computer with an arrow at the top.
Left or right?
So at the if you just drag your mouse over your box, then the options that come down at the top.
Okay.
Or toggle mic, toggle video, and then there should be share screen at the top there. Not seeing that?
Toggle full screen. I don't see share screen in my stuff.
I see full screen. Okay, that's something that we're going to troubleshoot this offline, because there may be a operating system or a computer kind of reason for this. So I'm just going to ask. Sorry about this, Arthur, but we'll have you up.
Okay, no problem.
So we're just going to bring up Alexandra. So Alexandra, I'm going to invite you up. Arthur, I'm going to remove you for a little bit, and I'm going to do some reconnaissance in the background to figure out, try and troubleshoot your problem, but try and get the session carrying on nevertheless.
Okay, I think I saw it. I think I think.
Hi, Alexandra.
Hi.
So can you just try and share your screen with me? And
yeah, sure.
So if you just hover over your face.
Yeah. So I got that. Yeah, it shows up right when I go over my face. Yeah.
Okay.
So you can see my screen now, right?
I can see your screen, and actually, for some reason, I can't see anything else right now.
Yeah. Oh, maybe I might need to click the close message.
Okay. So Arthur, just double check one more time. So Arthur, you don't see an option. So you see toggle video, toggle mic. You see that menu?
I see toggle HG, toggle video, yes.
But you don't see share screen in that menu. Okay, so Arthur's just removed himself for now, which is fine. Arthur, we'll have you back up in a bit. Alexandra, will have you kick off now and get things moving.
Okay, so let's try this again. So I share my screen. click to close message.
Okay, good. So I'm gonna.
All right, now this is working fine.
Focus. Yep. Okay, you're good to go.
Right. Okay. So let me just full screen this. I guess that's a bit better. Okay. So thank you, everyone. I guess this was somewhat an unconventional start to this presentation, but let's get on with it. So I'm also with André Dantin, and the work I'm presenting is jointly done with himself and Jakob Macke, who's now recently moving to Tübingen in Germany. And the basic topic is on inferring effective population models from data. So as probably most of you know, it's not so difficult to simulate from thousands of neurons at the same time, but then you end up with thousands of spike trains. And it also might be that your data that you're trying to model is not necessarily at that level of granularity. And for example, if you have like LFPs, then it makes more sense to think about populations. So you might want to model populations directly. And one other advantage that you have when you model populations is that you only have maybe tens of parameters rather than thousands. But you still need to choose those parameter values, and that's not a trivial task, as anyone that's tried to get simulations to look like data probably knows. And so I'm going to talk about techniques that we've developed for doing this process of going from some recording of data at the population level and back into inferring the parameters. So the basic setup that we have is that we have a bunch of mathematical equations that looks that will, when you feed them parameters and you integrate them forward in time, you get out a bunch of data that might correspond to population rates or something like this. And what we would like to do is find what those parameters should be given some data, and we want to do that in two possible different ways. So one would be gradient-based optimizers, where you this is a kind of thing that you might have in like SciPy or some other routine where you go up the gradient to maximize some cost function, or you can do something more Bayesian where you find the entire correlation structure between your parameters. And so what we need in order to do this is we need the likelihood of the parameters. So this is a combination of the data and the simulation parameters. From there, we can compute the likelihood. And once we have the likelihood, we take the derivative with respect to our set of parameters to then obtain these quantities on the right. And what of course, if we were to do this by hand, so if you try to compute the derivative all the way back to the initial, all the way back to your parameters by hand, that would be quite a bit of work. So we use tools that we develop for deep learning, where you differentiate through multiple layers of a deep neural network. But instead of differentiating through layers of a network, we differentiate through the integration steps of our simulator. And so this is what I'm showing over here. With your deep learning framework. So by deep learning framework, I mean something like PyTorch or TensorFlow, which you may have heard of. That caveat means that we can't just use something like an off-the-shelf neuron simulator, and we have to build our own. So that was quite a bit of work, and we now have code that we've put out to kind of help that process and make it more reliable. Okay, so once you've got this framework, this setup where you can infer parameters from a simulator on top of a model, or given some data, what can you do with it? So if you were to imagine that you only had like a generic black box neural network, this thing can map almost any input to any output. So you could definitely train it on something like a sinusoidal input and expect it to learn what to output as a realistic network. But then if you change the kind of input, the training input, you would also, you have no idea of what's going to come out. Whereas in this case, we're fitting the parameters of the set of equations that were designed specifically to map, to reproduce neural activity. And so by doing that, by doing that, we basically have much more robustness in terms of the fit, and we have better generalizability. So what we did was trained here on these sinusoidal inputs, and by testing on a very different kind of input, we still get very good match between our kind of full model, where we took every neuron, our population model, where we took the averages, and the effective model, where we took the inferred parameters that we got from this optimization process. And the reason these are all really cool is because the simulation we did here is with, they actually had homogeneous populations. So the theory where you use the mean parameters is actually a very good theory, and you get very good match between the two. But of course, neurons are not homogeneous, so populations are all different. Every neuron is different. And so if you go in and you redo the simulation with different parameters for every individual neurons, then the match between this homogeneous kind of population model is not as good with the true with the underlying kind of true simulation that we're using. And that might suggest that your model is not really good enough for your data for this kind of data. But that's not so what we found is that if you keep the same equations, but you just, instead of taking the mean parameters, you let this inference procedure find better fitting parameters to your data, you can actually get something that's a lot better. So in this case here, it's exactly the same set of equations, but in contrast to before, the parameters that our optimizer finds are not going to be the same as a mean parameter. So they're going to be in slightly different values that compensate to some extent for the mismatch between the model and the data. And so we can bridge at least partially the gap with our model. And I'll just kind of wrap up in a single slide. The first thing we did was that we built this tool for building differentiable likelihoods from complex models. And this allows us to fit these models to data. And then we showed that the effective parameters that we find are able to generalize to novel inputs, and this is due to the structure of the equations. We found that the effective parameters are also able to compensate between differences between the model and the data. And something I didn't talk about, but we can also do Bayesian inference with this. It's the same because everything is still based on the likelihood and the gradient of the likelihood, so we can find these kind of correlation structures between different parameter sets. And with that, I'm going to already conclude. So this work is already available on arXiv for a number of months already, and you'll see it in Continual Computation in a few weeks. And I'd like to thank all the people and all the institutions that have helped me over the last few years. Okay. I guess there's one I could ask for questions, I guess, at this point.
Yeah, so we'll move pretty quickly because we have to get through and we had a short start. But I asked the question, and I'll read the question. So what AutoDiff framework are you using?
So in this case, we're using Theano, mostly for historical reasons, because this work started at a time where Theano was still the most capable of these frameworks. It is, to a large extent, agnostic. So there's an agnostic layer between the AutoDiff framework and the actual modeling. And so, I mean, at some point in the future, we're going to have to change that. And so having that layer is going to make that easier to do. But yeah, we're using Theano for now.
That's great. I actually have been playing around in this area myself. I haven't been doing it nearly as systematically as you have, but this idea of, letting AutoDiff solve the problem of finding Jacobians is, it seems like a very good idea.
I would think so, yeah.
Yeah, and so I got some good initial results with AutoGrad, but I noticed it's relatively new. But if you go to the AutoGrad webpage, then now they're focusing on a new library called JAX.
Yeah,
which is where all the action is going to happen. It looks like.
Weirdly enough, JAX is, I believe, philosophically closer to Theano than TensorFlow and PyTorch. They kind of went back to the full functional setup, which makes the definition of the chain rule through the derivative somewhat more consistent and robust, I think.
Interesting. Okay, definitely going to check out the repo and follow this work, and it definitely addresses a major need we have in the field, which is being able to fit models. Yeah. Excellent. So thanks a lot, Alexandre, I guess.
Yeah,
and
but yeah,
we'll move on to the next talk.
Cool, thanks.
Boot you off from the screen. Now, Arthur, let's have another go. I'm going to, if you're still there, Arthur, I'm going to bring your screen back up. So can you...
I go to toggle video, right?
Okay, so next to toggle video, there should be an option to share screen. Okay. So it's not toggle video. It's like next to toggle video. I guess you're not seeing it. You know what I'm going to do? I'm going to disinvite you and then re-invite you and see if that. Oh, okay. Yes. Amazing. Fantastic. Okay, so you have your screen being shared. So if go to your slide presentation tool of choice.
Do you see my slide?
Yes, I do.
Okay.
Okay, so Arthur, take it away.
Okay, thank you very much, John. So I am Arthur Wanwe from the University of Ottawa in the Downtown Group. So I will present my work, Dynamic and Statistic of Gamma and Beta bursts. So let me explain first what I mean by gamma bursts. So these are LFE recorded from the primary visual cortex of a monkey. And you can see below the corresponding time frequency representation. You see that oscillation here appear as short and random epoch of synchrony. It is this short and random epoch of synchrony that we call gamma bursts. Gamma because we are in the gamma frequency range, so between 30 and 100 Hz. So this kind of signal really are closer to a filter noise than to well-coherent oscillation. However, we can still characterize them by just looking at the duration of the burst and the corresponding frequency content. And that's what is done in this panel. So the mean of these bursts are typically around 100 milliseconds. But this kind of behavior is not just seen in the monkey. You can also see this kind of behavior in the human or in the cat, or in other frequency band like beta, for example. So our modeling work here aims to first characterize dynamically gamma bursts, match the dynamic of bursts to biophysical parameter, and compute mean burst duration. So we start with a stochastic Wilson-Cohen model. So the model has a deterministic part and a stochastic part. So we will be mostly interested in the parameter regime where this deterministic part has complex conjugate eigenvalue. So the top panel here are bifurcation diagram of the model for the synchronization level and the frequency respectively. So you see here that the line in black here is the up bifurcation. If you are in this area, you have limit cycle oscillation, and a point here in black will lead this kind of self-sustained oscillation without any burst. However, if you are in this area, oscillation are actually induced by noise. So without noise, you do not have oscillation. And the black dot in this area leads to this kind of gamma burst oscillation similar to what we actually saw in vivo. And you have the corresponding LFE in the bottom, and you see that bursts actually match with the epoch where the amplitude of the process is high. So we will focus in this domain of the parameter regime and will not care about the limit cycle area. So we will further add 3 other points here in red, depending on their distance to the bifurcation, to the up bifurcation. So the goal is actually to find the dynamic of this amplitude envelope in black. So we apply linear noise approximation and linearization on the stochastic Wilson-Cohen model. I mean, you have the dynamic of the equation here with the parameter that depend on all other network parameters. We look for the solution of this equation in this form, and we replace them in the linear equation sustained by node that I just showed, and we apply the stochastic averaging method. Then we have the dynamic of the envelope there and the dynamic of the phase. So the dynamic of the envelope here just depends on 2 meta parameters, and more importantly, the dynamic of the envelope is coupled with the phase. So we can much easier compute mean burst duration using this dynamic. And to compute mean burst duration, we first define a burst like the epoch where the amplitude is sustained above a threshold that we have chosen. So here in magenta. So the mean burst duration will match with the time that the process go up and down. And we use mean first passage time analysis to compute these 2 duration. And we just add them together to get the mean burst duration. So this mean burst duration is given by this relation and depend on all network parameter through the 2 meta parameter and also to the distance to the up bifurcation. Now we examine the case depending on whether you are far or close to the bifurcation. So the first panel here correspond to a point very far from the bifurcation. So we see that the mean burst duration computed is 35 milliseconds, so really shorter than what is actually seen in vivo, which is around 100 milliseconds. And for a point too close to the bifurcation, we found that the mean burst duration is 550 milliseconds, so very higher than what is saw in vivo. But in the middle, we have mean burst duration, which are actually in the range of what is actually found in real data. So we assume that there exists an optimal range of parameters which match in vivo data, and deviation from that range actually lead to pathological case. So I end my talk by just insisting on the fact that future work should try to integrate external stimulus with a special attention to deep brain stimulation. So thank you very much. So if there's any question, I am open.
So thanks a lot, Arthur. And I have a question, which I'm actually just going to type so that we have it as a record, but I'll read it while I type as well, which is, what are the considerations? In fact, give me 1 second and then I'll ask you to answer it. What are the considerations using Lucid? Okay, so the question is, so you're using a stochastic Wilson-Cowan model?
Yes.
Yes. And so what are the considerations in terms of, say, like this bursting behavior with the stochastic version of the model versus a, deterministic, but maybe like driven by a periodic driving input, for example?
Yes. As I show, maybe I can show you again with my. As I show here, if you have like a deterministic Wilson-Cowan model, you just have limit cycle oscillation. So you will be in this regime, and you will have this kind of oscillation. So you will not have bursts in your dynamic. You will not have bursts in your dynamic in a deterministic regime, because in this part of the parameter regime, the dynamic will go to the fixed point. So we don't have any dynamic here. And if you are in the limit cycle part, you will have just sustained oscillation like this one. So you will not have this kind of bursting dynamic. So to have bursting dynamic, you need to have stochastic part in the Wilson-Cowan model, and you need to be in this regime. So that is, I don't know if I answered to your question.
Yes, you answered perfectly. That was great. So we don't have any other questions, and we have 2 other speakers, although we're not in a huge hurry. By the way, Alexandra, I'm sorry I maybe moved you along quickly. We only need to be somewhere at 5 o'clock. That's when the wrap-up time is. So we have 4 speakers, I believe. The next 2 are here, and I'm going to open up the stage to our 3rd speaker. So I just want to, let's have a virtual round of applause for Arthur for that fascinating brief talk. Thanks a lot, Arthur. I'm going to give you the boot, and we'll hear from...
Thank you very much.
You're welcome. So we have Golia and Shabab. Now, Golia and Shabab, do you remember which one of you is up next? I don't have the schedule in front of me right now. Or do you want to fight over it? Either's fine. Okay.
Okay, hi.
Wendy. Hi, Galia.
Hi, how are you?
Excellent, thanks. Excellent. So hopefully we can achieve screen sharing with you quickly.
Yeah,
so if you
sure.
Over, you should have an option to share screen.
Yes. Okay, can you see my screen now?
I see your screen. I'm enlarging your screen.
Thank you.
And I'm going to mute my microphone, so take it away.
Okay, thank you so much. My name is Golia. I'm a PhD candidate at McGill University, working in the neuroscience department with Dr. Bratislav Mizik. So today I'm going to talk about hierarchical organization of local brain dynamics across the human cortex. So in our lab and in this project specifically, we study the brain as a complex network. And for this, the first thing we need is some brain imaging data. We then apply any sort of parcellation to this data to subdivide the brain to a number of gray matter parcels or nodes. We can then use techniques such as diffusion imaging or tractography to construct a graph model of the brain where the nodes of this network are brain regions or parcels, and the edges are the white matter anatomical connections between them. This information can then be summarized in a structural connectivity matrix where rows and columns are regions or nodes, and the elements are the connections. We can also use functional MRI or functional imaging to measure brain activity over time and use the resulting time series to quantify the functional interactions between pairs of brain regions and basically construct this functional connectivity matrix. So this graph representation of the brain is one of the most common approaches that people use to study the brain as a complex system. However, it lacks information about regional heterogeneity across the cortex. So for example, if we take node A from visual cortex and node B from frontal areas and have a closer look at some of their local properties, such as morphometrics, cytoarchitectonic, transcription, and temporal features, we see that these features could be highly similar or drastically different for these two areas. So there is this variation of properties across the cortex. And one of the interesting things about this is that the distribution of most of these properties show this separation between regions that belong to lower order unimodal sensory regions and the ones that belong to higher order transmodal areas. So a lot of people have tried to relate local properties to a large scale network organization of the brain. I wanted to do basically the same thing, taking the same approach, however, mainly focusing on local cortical dynamics. So for this, we use feature-based representation of time series, where we have fMRI time series of the whole brain. We use a CTSA to extract a large number of features from this fMRI time series and basically quantify a temporal profile or a dynamic fingerprint for brain regions. We can then do various things with this data, such as we can see how correlated this temporal profile or this dynamic fingerprint is across the cortex and construct a temporal similarity network where the elements show how similar these temporal properties are for two brain regions. We can also perform dimensionality reduction techniques such as PCA and look at the lower dimensional projection of these temporal features across the brain. So we first construct this temporal similarity network, and we look at this in relation to other large-scale other properties of large-scale brain networks, such as Euclidean distance. And what we observe here is that the areas, the regions that are closer to one another have a higher temporal similarity. So their temporal properties are similar to each other. Also, the temporal similarity is significantly higher for regions that are connected to each other with the structural connections or for the ones that belong to the same intrinsic functional network. If we directly compare temporal similarity to functional connectivity, we see this positive trend showing that the stronger the functional connection, that more similar temporal properties. So basically, altogether, this shows that regions that are closer to each other, that are connected to each other with an anatomical connections, or that belong to the same functional system have similar temporal properties. So we next apply principal component analysis, or PCA, on this data to identify a linear combination of temporal features with maximum variation across the cortex. So with this, we want to look at the spatial distribution of the dominant dynamic profile of fMRI, of features of fMRI time series over the brain. And we see that the first component kind of shows this rostrocaudal gradient starting from visual areas going to more like mediotemporal regions. And the second component separates unimodal sensory areas from higher order integrative regions of the brain. We directly compared this to other measures of hierarchical organization of the brain, such as principal functional hierarchy, which is this hierarchy of functional connectivity. And as expected, we see that our second component is significantly correlated with the functional hierarchy. We also look at the principal component of gene expression, which is basically the dominant spatial variation of gene expression across the cortex. And we see that both of our components kind of reflect this gradient. And altogether, this shows that the gradient of temporal features across the cortex is in line with gradient of other characteristics of the brain. And we also attempted to interpret the features, so identifying the features that contribute mostly to the identified pattern and to this dominant dynamical fingerprint using univariate analysis of PCA loadings. And what we observe is that the measures that are mainly contributing to the observed patterns are the ones that have to do with the autocorrelation of the signal and with the entropy or the variability of the signal, which are some measures that make sense in the case of fMRI or functional MRI. So in summary, we see that dynamic fingerprints of regional brain activity is related to the macro scale network embedding, and pairwise similarity of these dynamics reflect brain network topology and geometry. And these dynamics reflect molecular and cytoarchitectonic gradients. And we also observe this hierarchically organized dynamics across the cortex that recapitulate the unimodal transmodal hierarchy. And with this, I want to thank the lab, my PI, the collaborators, the funding agencies, and thank you for listening.
Thanks, Galia. Really excellent work.
Thank you.
Yeah, fascinating and really neat idea as well to take the highly comparative time series, data as a feature and then compare between regions. So I had a question to kick off, which is, so people who maybe aren't so familiar with this method, Ben Fulcher's method, highly comparative time series analysis, you get how many thousand features can you get from lots, right?
Yeah, so it's a lot of features. So we can take over 6,000 or 7,000 features for each region.
Right.
Yeah, most of them. So they could be like very simple features, such as the mean or the median of the distribution of the values of the time series, or there could be more complex, such as measures of autocorrelation, measures of like entropy, automutual information, that type of measures.
Yeah. So, and you did actually answer this to some extent, I think near the end, but to ask it again. So, did you use all of the features, like the full stack? And within that full stack, what types of, properties are primarily represented? You mentioned autocorrelation, and yeah.
Yeah. So I tried to keep all the features. So, but we first performed like a normalization step to keep the features that are robust and that makes sense. If we had errors or like outliers or those things, so we corrected for those and we kept the normalized features. But at the end, we ended up with like about 6,000 features instead of 7,000 something, but still a lot of features. And then using this type of univariate analysis, we found that measures that have to do with autocorrelation of the signal. So basically, how similar the future values of a signal, of brain signal can be predicted from the previous values. Basically, it's like measure of autocorrelation, but also measures of entropy. So how variable, so how noisy our signal is kind of. So these are the ones that we found that contribute mostly, and that weight heavily on our PC loadings that kind of contribute also to these spatial variations.
Great answer. Another two quick ones, both from me again, but I kind of know this area, so I'm indulging myself. And,
of course.
The audience has been quite quiet, I have to say, Andre, I know you're there, Andre, let's have some cues, Dimitris, but anyway, so the one simple question is, you mentioned a principal functional hierarchy. So is this the principal gradient, Daniel Margulies?
Exactly.
Connectivity principal eigenvector. Okay.
Yes, exactly. So we use diffusion map embedding following exactly Daniel Margulies's method on HCP resting state MRI data. And we kind of identified the same gradient that he reports in his paper, in his original 2016 paper, but in HCP datasets.
Great. And second, and I say final question from me is, and I'm sure you've done this, is you've been focusing, or we've described so far, as the variability over regions, right? But I can't remember if you showed it, but you've got variability over subjects as well. So which is higher? I mean, is there is everyone got a somewhat similar feature vector in the visual cortex, or is my visual cortex feature vector more similar to my frontal cortex feature vector than my visual cortex feature vector is to the next guy's visual cortex feature vector?
Right. So what we did, we actually wanted to know the exact same thing. So what we did was to perform the same analysis, but on subject level basis. So basically performing PCA and extracting features for each subject. And after aligning, all the our PCA analysis to one another for all the subjects, we ended up seeing that most of the subjects, they have this same distribution across the cortex. So if you look at the average distribution of the features across the cortex for all the subjects, you see the same pattern as if you only use the group average data, which is what I'm showing here. So basically, I'd say this is some maybe a general measure of, I'd say, there is, of course, individual variation, but if we are looking at this spatial distribution of the dominant kind of pattern, I'd say it's kind of a general maybe feature that we see, the same as what, for example, they see for functional hierarchy.
Fascinating. Well, I'll leave it there on the questions and just take the opportunity to thank Gulya again for an excellent talk and some fascinating work. And now I'm going to ceremonially give you the boot and invite up Shahab for the final talk of the session. Thanks again,
Thank
Gulya.
you. Thanks, bye.
Okay.
Hi. Hey.
Shahab, hello.
Hi. Do you hear me?
I hear you fine.
Okay, great. Okay. Nice to see you.
Nice to meet you. So if you could share your screen.
Sure.
If you just hover over and you should see a tab in the menu. Yep, very good.
Okay, you see my presentation now?
See your presentation. So I'll make it big and take it away.
Okay, thank you. So hi, everyone. My name is Shahab Bakhtiari. I'm a postdoc with Blake Richards at Mila in Montreal. And I'm going to talk to you about some preliminary results that we have on this project, where we are trying to develop an artificial neural network model for mouse visual cortex. So if I just wanted to put it very broadly, the goal of this project is to develop an artificial neural network model that has similar latent representations to mouse visual cortex, a similar pattern of neural activity stimulated by natural images and videos. So I'm sure that you're all familiar with this study from DiCarlo and his team. So in this project, DiCarlo and others tried to basically train a deep artificial neural network model to do image classification or object recognition. And what they realized was that this artificial neural network, after being trained on image classification, the representations that are learned across different layers are similar to representations in the ventral pathway of monkeys' visual cortex. So basically, different layers of these models are good predictive models for the activity of different layers of processing along the ventral visual pathway. So this study gives us some general idea of what this machinery, this multi-stage machinery of ventral pathway is implementing at a computational level, and it was very helpful for the whole field of vision neuroscience. I think it was one of the most successful marriages of deep learning and vision neuroscience. So, given the recent relatively recent interest in studying the visual system of mice, having such model would be very useful for both experimentalists and theoreticians. So this is basically the goal that we're after in this project. But as soon as we started working on this project, we realized that image classification or object recognition as a cost function is not very optimal if we are trying to model mouse visual cortex. And the reason mainly is that, first of all, not a very great portion of mouse visual cortex is developed and evolved for object processing and object recognition. And second of all, and I think more importantly, what a mouse would call an object in a day-to-day basis is different than what we humans would call objects. And in the way that, for example, a data set like ImageNet is annotated with objects. So we realized that we need a more general type of objective function or cost function that is also relevant across different species. For this reason, and in this project, we started looking at temporal prediction as a cost function. And we took a simple deep artificial neural network, and instead of training these models on image classification, we trained them on temporal prediction of natural videos. And basically, what the model is trained to do is that given the present frame of a video and the past frames, the model should be able to present the next frame, predict the next frame of the same video. So the model is trained to do this predictive task, and what happens is that then we go and look at the representations learned across different layers of these trained models and compare the representations with those in mouse visual cortex, different areas of mouse visual cortex. So we in terms of analysis, we did something called representation similarity analysis. I'm sure that many of you are familiar with this framework. I'll not go into the details of this, but in short, for those who are not familiar, we are looking at a measure called RSM similarity. So first, we have a matrix called representation similarity matrix that quantifies the representation of a natural video either across different layers of our trained model or in different areas of the mouse visual cortex. So we have this RSM matrix both for different layers of our model and for the animal brain. And then we look at a correlation measure of these RSM quantifications. So for example, we have RSM for our artificial neural network model. We have an RSM for V1, for example, in mouse. And we look at this correlation and we call it RSM similarity. And this is basically the value that tells us how well our model is capturing the representations in the mouse visual cortex. Based on that quantification, we get a plot like this for example, for area V1. The vertical axis shows RSM similarity, the value that I just described to you, and the horizontal axis shows different layers of my trained model. And this is only for area V1. And what we see, for example, here is that for layer 8, we are getting to a value around 0.4, which is around 65% of explainable representation, given the intrinsic noisiness and variability of responses in vivo recordings. And so we can now take this maximum value of RSM similarity and compare this maximum value with the maximum value that we would get from an image classification trained model. So this is a type of comparison that we get. The brownish bar here shows the R model, the maximum RSM similarity for R model, and the blue one shows an image classification trained model for area V1. And we can see that our model is doing a better job compared to the image classification model. We can do the same for all the secondary visual areas. Here I'm showing the lateral visual areas of mouse visual cortex. And you can see that, for example, for VIS-AL and VIS-RL, two lateral areas, our model is doing a better job, and for VIS-L is doing almost the same. One important difference that we saw in our results was that in lateral areas, we are relatively doing a good job. Our temporal prediction model is doing a good job. While we look at the medial areas, we don't see any difference between our model and image classification model. And also, it seems that for area PM, VIS-PM, the image classification is doing a bit of a better job. So this is the preliminary results that we have so far, and now we are working on different ways of improving the similarity of representations. For example, incorporating different cost functions in the same model, mainly in order to get better representations for medial areas. And also, one important step is to make the architecture of the model more similar to the mouse visual cortex, which is not really a purely feed-forward architecture. And on that note, I wanted to thank all my collaborators, team members, and especially Blake Richards, my supervisor. Thank you all.
Thanks a lot, Shahab. That's really fascinating work. It's a really nice idea. I have a question, and I'm gonna ask a question, but first I'm gonna ask for a clarification on the methodology.
Sure.
And that's so you're training your classifier to predict the next frame each time.
So there is a bit of some subtlety there. Our model is not really reproducing or reconstructing the next frame. So let me take you to the slide here. So the model is not really reconstructing the next frame, but rather what it does is doing a classification task. It is solving the task that either the frame that we fed here through the network, is it the right next frame or is it the wrong next frame? And we call this, this is a very famous method called contrastive noise estimation. So the model is basically a classification that learns the sample that is fed to the model is coming from a positive distribution or a negative distribution. And it makes the training a bit easier because you have to deal with less number of parameters and so on.
Okay. So thanks for the clarification, and perhaps it helps with, perhaps it matters for my question, or perhaps it doesn't. But the first thing that came to mind is that because you're doing something or other that's like frame to frame, based, then you would be capturing less kind of conceptual abstractions than the type of things that Nico has done with object categorization, which are highly visually non-similar. That's kind of the whole point of abstract conceptual abstractions, right? You could have something that's visually similar but it's the same, or visually non-similar but vice versa, not that, the opposite. So yeah, like using categorizations would allow more for abstractions than using frame-to-frame similarity predictions. Is that broadly correct?
So it is correct. If we wanted to predict the next frame at a pixel level, that would be definitely true. But we are not really predicting the next frame at a pixel level. So if you look at this graph, we are actually predicting the next frame at the same latent level as the output of our convolutional neural network. So you would assume that the convolutional neural network is forming a latent embedding that is more abstract than the pixel level representation, and we are trying to predict the next frame in the same latent embedding. And so basically this is a difference here.
Yeah. Okay. So I kind of missed the point. I think.
That was my bad. I couldn't go into the details. Yeah.
It's really fascinating approach. Do we have any other questions in the tab? Okay, we don't have any other burning questions. Okay. So I think we'll leave it there. That's been some really fascinating work from our 4 speakers. So I want to just extend my thanks and congratulations to all of you. Thanks again for presenting to us. And once again, so Shahab, nice to meet you. Great talk. I'm going to give you the boot.
Yeah, thank you very much. Have a good day.
And to everyone in the session, so we have just under 10 minutes to chill out and get yourself a cup of tea or something. The wrap-up closing remarks for the day are going to be held at 5. So you can just shuffle over to there at 5 o'clock, and we'll see you there.